Hey everyone, Jack Audie here. This video we're going to jump into the innate immune system, which is a branch of the immune system, one of the two major branches. And the reason why I'm jumping into this is because the innate immune system branch is going to be the first branch that is exposed to SARS-CoV-2. So I want to teach you guys about how the immune system responds to SARS-CoV-2 or COVID-19. So a good place to start is the innate immune system, which is the first immune branch that will meet the virus. So let's jump into it. What have we got here? Okay, so the rough organization of the immune system is we have barriers, which is things like our skin. It's kind of boring, but it is important. The dead outer layer of our skin is an amazing protective agent, a protective mechanism against pathogens. And this is why if we get a cut or a blister that breaks the surface of our skin, it opens us up to infections. So barriers are doing a lot of work when it comes to protecting us from infections. Next, we have the innate immune system, and I like to think of the innate immune system as a bit like a tank, and that's because it is non-specific. It uses relatively crude mechanisms to wipe out the pathogens, and in doing so, it can also cause collateral damage. So some of the techniques the innate immune system uses, because they're non-specific, damage both the bacteria and damage us too. There's a couple of them, but I'll go into it. Whereas the adaptive immune system is a little bit more like the FBI. It does a targeted approach to a specific threat, and it takes out that threat whilst doing very little collateral damage. Now, both the innate and adaptive immune system have sort of a protein part that floats around our blood and a cellular part, which is cells roaming around taking out pathogens. In the innate immune system, it's the complement. Now, I'm not going to go into this hugely, but basically the complements are a group of proteins in our blood that can spontaneously activate in the presence of a pathogen, and once activated can do mechanisms to kill that pathogen or at least help your cellular immune system deal with that pathogen. For example, complements can poke holes in bacterial cell walls to lyse the bacteria. Your adaptive immune system has humoral immunity, which is just antibodies, which are proteins that float around your blood, and that they are specific, and they will specifically bind to one pathogen, and they can take out that pathogen through a number of means, but one is just smothering that pathogen. But the antibodies also aid the cellular component of both the adaptive and innate immune system to take out that pathogen. So that's the rough organization. Let's jump into the innate immune system. Now, I've broken down the innate immune response into four main branches. You've got sensing the pathogen or tissue damage. You've got signaling. So then after it senses the pathogen, it must signal for the immune response to occur. Then you have the immune response, which includes both a vascular and a cellular component, a blood component as well as a cell component, and then you have to resolve that immune response back to normal. So we're going to quickly run through this. Let's have a look. So, so the innate immune system is responsible for a process that we call inflammation, but inflammation is just an acute innate immune response to tissue damage or infection. So here we have our immune cell sitting in tissue. So the brown cells are just a regular piece of tissue. It could be your skin, it could be anything. We've got a blood vessel running through here, and here we have an innate immune cell just sitting in our tissue. There's a few innate immune cells that sit in our tissues. There's dendritic cells, there's mast cells, and there's also macrophages. This looks a lot like a macrophage. It's much bigger than all the other cells, so this is probably a macrophage or the big eater. Now it's just sitting in there, and what it's doing is it's surveying the environment. Then something's going to happen. Now, let's say this is a sterile injury. There's no pathogens. We just get tissue damage. What's going to happen? Well, with tissue damage, you're going to get cells popping, and then chemicals that normally sit inside the cell are going to come outside the cell. So normally there are chemicals or compounds that are only present inside of cells. When cells pop, they will release those. And some of those can act as what's called DAMPs, which is a damage-associated molecular pattern. DAMPs, right? And so these DAMPs will come out and they will be detected by a range of detection molecules, receptors, on the surface of the macrophage or even inside the macrophage. So that's one possibility of sensing. Another one is PAMPs, which are pathogen-associated molecular patterns. And these are compounds that are released or on the surface of pathogens. So here we have a bacteria here, and they are releasing a PAMP into the extracellular milieu, and that is binding to receptors on the macrophages. So after receptor activation, after they have sensed the pathogen or tissue damage, they will then go into signaling. Now, there are loads of signaling molecules, but here's just two examples, prostaglandins and cytokines, and I'm going to do a deeper dive on those in the next video. So an immune cell that's resident in the tissue, like this macrophage, will detect tissue damage or a pathogen, and then will release signaling molecules like prostaglandins and cytokines. These signaling molecules will go bind to receptors on other cells to initiate the immune response. So the first one is vasodilation and leaking. So the blood vessel, the signaling molecules will act on receptors on the blood vessel, causing it to expand, and for the bonds in the blood vessel cells to break down, making them leaky. Why do you want to make them leaky? And why do you want to vasodilate them? Well, you want to vasodilate them to get more blood to the region. You want to make them leaky so the protein components of the immune system can now leak out of the blood and get into the tissue. So the complement proteins that I talked about in the innate immune response can now get into the tissue, but also the antibodies that are present in the blood may now flow into the tissue where they might bind and do stuff to the pathogens. So that's why you want your blood vessels to get leaky. And this is why your ankle swells up when you roll your ankle. Now your blood vessels are dilated and fluid is rushing out, and so your ankle swells up into like a big water bomb kind of thing. Then what's going to happen is those signaling molecules are going to go and act on immune cells that are then going to be recruited to the site. So here we have a few examples. We've got the neutrophil. Now I'm going to go into detail on each of these cells in a later video, but we've got, say, for example, the neutrophil. This is actually most of the white blood cells in your body. The neutrophil will recruit in, and it will start releasing pathogen killing compounds. And one example of this is neutrophils actually produce bleach. Now, and they produce bleach to sterilize the area, exactly the same reason why we use bleach in our kitchens. And so when a neutrophil produces bleach, that bleach will then react with the membrane of the pathogen, for example, and the membrane will become leaky, and the cell and the pathogen will now leak and lyse and die, right? But the problem is the bleach is doing the same thing to our tissue. So this is why we say the innate immune system is non-specific, because that would kill all pathogens. Bleach will be great at killing viruses and all kinds of bacteria and fungus, for example. But also that it's a bit cruder, because instead of just taking out the pathogen, it can actually take out peripheral tissue, which is maybe one of the reasons why we ice our ankle, to try and reduce that collateral damage that's going on. Now, we also recruit monocytes from our blood. Monocytes are a lot like a macrophage. They will come in and they will take over managing the immune response by releasing prostaglandins and cytokines. And other signaling molecules, but monocytes can actually also then turn into resident macrophages. So they can replace the macrophages that might have died during this innate immune response. And then we need resolution. So after that, after the inflammatory signaling molecules are released, that actually triggers the release of an immune suppressing anti-inflammatory molecules to lead to resolution. And so we need this immune response to stop. It can cause a lot of problems if this immune response continues. And so we need to head to resolution, and that occurs through signaling molecules. In response to the inflammatory signaling molecules, we get anti-inflammatory signaling molecules that turn everything off, and they even promote tissue growth and revascularization. If a blood vessel was damaged, it promotes revascularization and new blood vessel forming. So we need that resolution to occur. Now, there's one little side track that I haven't mentioned of the innate immune system, and that's the natural killer cell. So the natural killer cell sort of operates funnily enough, as a little assassin by itself. It's less part of that team, and it more is a lone assassin. And it flows around the blood. It's predominantly in the blood, and then it will be recruited to the site through those inflammatory signaling molecules. Now, the great thing about the natural killer cell is it is basically programmed to destroy. It is thinking, I am going to kill every cell I come in contact with. So here is a natural killer cell, and it's going to approach a lung epithelial cell, and it's going to try and kill it. But then what's going to happen is there is a receptor on that lung epithelial cell, and that receptor is called MHC1, right? Now, MHC1, this receptor, what it does is it displays the proteins from within inside the lung epithelia. So if there are normal proteins inside the lung epithelial cell, or any cell for that matter, so all cells express MHC1, so any cell in your body will be expressing MHC1, and on that MHC1 will be a fragment of the normal proteins sampled from within that cell. Now, the natural killer cell will go to kill this lung epithelial cell, but then it will bind to this receptor and detect it, and it will say, hold on a minute, I am seeing a normal human protein on the surface of this lung epithelial cell. I've checked its MHC1, it contains a fragment of a normal protein, and this inhibits it from killing it. So basically it says, whoa, 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 don't kill the lung epithelial cell. So the natural killer cell then thinks you will not die today. Now, if this lung epithelial cell was a cancer cell, it might have aberrant proteins, or if it was infected with a virus, it will have viral proteins. So on the MHC1, it will express a fragment of a viral protein, and this will not be recognized. So the natural killer cell will go, that is not a human, that is not me, that is not my specific human milieu of proteins, it's not my little fragment of me. And so there will be no recognition, so there won't be the inhibition. So what will then happen is it's granule time. Basically, the natural killer cell is full of granules, and the granules will then fuse with the membrane of the natural killer cell, releasing the contents of the granules. Now, in the granules, it's a complicated mix of stuff, but one example of what's inside it is perforin. Perforin is a perforator protein, so it creates holes in the proteins, it creates holes in the membranes of the cells exposed to perforin. So what it will crudely do is these little, I've crudely drawn them as bean-shaped proteins, will insert themselves into the membrane of the neighboring cell, and they will form a tight little ring in that membrane, creating a hole. Now, this hole completely destroys the ion composition of the cell, and it causes a signaling cascade within the cell for it to die and undergo apoptosis. There are also other contents inside the granules of the natural killer cell, and those other proteins, such as gran enzyme, will go in and help initiate that apoptotic response. But the key here is that in there are killing compounds, cytotoxic compounds such as perforin, why can't I say that word? Such as perforin, that will perforate the cell. Now, I know that's a crude diagram, so I've actually got a fantastic paper that was actually created at Monash University, Australia, represent, Australia represent. I'm from the University of Tasmania, but anyway, we're all together. So they actually mapped the molecular structure of these perforin complexes, and so here we can see here is this is what it's forming in the membrane, and you can see it creates a nice hole, and here is a scanning electron microscope image of it, so we can actually see the proteins creating this hole in the membrane, which is such cool research. Very well done to that team. So next up, that was a big crash course in the innate immune system, just to catch everyone up, get everyone on the same page. Now we're going to do a bit of a deep dive about how that works, and that's coming up in the next video.